Oh, you both got pictures up.
Hey, Danny.
Hey, that's Jason from the
Internet?
oh, from the civil and electrical engineering.
Oh, are you doing your thing now?
Yeah.
Oh, sorry.
That's okay. You'll hear my kids in the background, so it's very informal. Hey, Eli.
Hey, Jason. Nice to meet you.
Good to meet you. Yeah. Is that right? It's Eli?
Yeah.
Okay. Okay. Let's see. Okay, so I appreciate. Or do y'all know each other first of all? Eli and
Yes.
Danny.
Yes.
Okay. Do y'all work in the same lab? Okay.
Yeah.
So I guess I appreciate your email, Eli, telling me what you wanted to learn, and I guess I think I'll just start at the very beginning, and then we'll kind of zoom in where we need more focus, and zoom out where we. So, can you see my terminal now?
Yes. Okay,
good. So here, I guess first I should show you. So y'all are working with corn, right?
What?
And so the first thing I did is I need to get a reference genome because y'all are interested now in applying your sequencing samples to the full genome. Is that correct?
Yes.
Okay. And so you need a reference genome to start with, and Illumina puts out a really nice reference, a group of references, and they call them iGenomes. Maybe they're ripping off of Apple or something. But down here at the bottom, you can see they have corn, but they only have the ensemble. So basically, ensemble is the European official repository for genes and sequences and stuff like that. And so I did a little more research, and it looks like there's also the B73 reference. So the ensemble one is available here, the AGPV3 genome assembly. But then maize GDB and plant GDB / maize seem to push the B73 reference. And I have no idea which of those is better. Do y'all have any prior experience with those?
I would recommend the reference genome 3 from
Okay.
B73.
Okay, so B73 version 3?
Yes, I think version 2 had a duplication error with the chromosomes.
Oh, okay. Okay, so then we need to find version 3, because this is version 2 here. This one, let's see what this version is. It says 1.20. Do you know where version 3 is available from? Oh, and just so y'all know, I'm recording this so that if y'all need to see it later or something, you can.
Good idea.
All right.
Okay, there's only version 2. Maybe they haven't released it.
Oh.
I mean, we can blast through the version 3 in maize GDB, but I haven't actually found version 3. Past the file version 3.
How do you know about version 3? I'm just curious.
Charles, what's the guy's name? Charles Johnson?
Yeah, Charlie Johnson. Yeah.
He mentioned it during one of the meetings I went to with him.
Okay. So you're sure it wasn't AGP version 3?
I'm not sure.
Okay. Well, we can just pick one and go with it for now, and then once y'all see how to do it, then you can, if you find version 3, then you can. So let's get version 2, or would you rather go with AGP v3?
Whatever's most convenient, version 2.
Okay.
As long as we can do it with version 2, we find version 3, we will do it.
Okay. So now I'm just finding out where's the FASTA file. And so I don't know what these FPC files are. Usually they're in FASTA format, so they'll have an FAA extension. I don't see it there. Where's the other place?
I found it once in MaceGDB, but I'm not sure where.
Wait, what'd you find?
I think I found it one in MaceGDB. How about that that dumps the no, maybe not. I don't know. Sorry. Found it
Okay.
once in MaceGDB.
Let's see, where was I? Okay. I would think it would be easy to download. Download. These sizes look wrong, but these names look right. Let's try that. So just grab that location, switch back to terminal window, and here I'm downloading the iGenomes AGP, but I'm going to cancel that because it's going to take 20 more minutes. So I'm not going to bother with that. I'll keep it there. And I think I'll put it over here. And wget is classic downloader. That was pretty small, so I doubt that's the right file. It is compressed. What is this? I think it's just, oh. How big is it? It's only 1.5 megs. I don't think that's right. Well, I'm trying to remember how big the others. It's only got 1 million characters in it, so that's probably not right. Man, why is this? I guess I'm used to working on like human and rat and mouse, where it's, you know, it's very easy to find everything. I'm sure y'all probably run into this all the time where, oh yeah, this is, I guess I downloaded the correct thing. It's only 1.3 million base pairs. But I read somewhere that corn is, you know, somewhere around 2.4 gigabase pairs. Is that right?
3,500 genes, 35,000 genes.
Yeah. It's roughly the size of human then, right? I mean, I know the number of genes, but I'm just talking about total length.
I think I found it. It's an ensemble, though.
But that's the AGP, right?
Yeah, that's AGP.
Well, let's just go with that one for now. I'm going to try this one. AGP v3, and let me just move this. Whoops. I just downloaded the index. Okay. I have to find the actual file. Okay, so you can either download it per chromosome, or you can just get the whole genome, which is what we want. I mean, we could get each chromosome, but then we would just end up putting them all together again. Let me delete that. Okay, so this should just take a few seconds. Although it looks like it's probably coming from Europe, so it's slow. It's speeding up. And then if we're careful, we'll check the checksum when we're done, which basically it checks to make sure that there's no, download errors or bit flips or anything like that. Okay, so then I guess first of all, do y'all have access to some kind of workstation or cluster to do these type of operations?
I have an account in WSGI HPC.
Oh, okay.
I think
Yeah. Yeah, that'll work. And you can use Galaxy on that. So if you want to use Galaxy, you don't have to do these, command line wizardry or whatever. But you almost always have to do some command line stuff anyway, because you can't do everything with Galaxy. And also, you have much more flexibility doing it yourself, because you can, the newest version of a tool comes out, you just download it and start running with it. Whereas Galaxy, you have to wait for someone to package it up, and you can do that yourself, but it's almost harder in some cases because you're taking the tool and then wrapping it with Galaxy.
What is Galaxy anyway? Is that like a GUI?
Oh, yeah, sorry. I just assumed you've seen it. So Galaxy, PSU, is a piece of software made primarily by Penn State University, and it's basically a web interface that you can use to interface with a cluster specifically designed for biomedical or bioinformatic type of workflows. And so you can do things like convert from one format to another, FASTA manipulation, filtering and sorting, operating on genomic intervals, doing statistics, regression. And so I would use this whenever it fits your need well, if you want, because it's it's designed for this. So it's and it's a pretty nice piece of software. They put a lot of effort into it. And there's a Quanti at the WSGI runs a Galaxy Instant, so you can work with your files on the WSGI cluster through Galaxy. So that's nice. I won't cover it here mainly because I don't know it. I know a girl that does like to use it, but it's a little clunky sometimes, or it's just not flexible, really. I mean, that's my main problem is, if there's one thing it doesn't do that you want it to do, you have to pull your data out and do that one thing and then put it back in the Galaxy, and moving it in and out is kind of a pain. But yeah, look into that later if you want to. Okay, so we got that. I'm going to move around a little bit. I didn't mean to put it in there. Okay, there you go. So I just have this, I just have this FASTA GZ file. You can see it's GZ'd from the extension. So I need to unpack it. And that'll, by default, basically delete the compressed one. You can keep the compressed one with the K flag if you want.
So this terminal that you're in, what software is this?
GNZip. Yeah, GNZip is just basically part of the standard suite of programs that's installed on almost any Linux computer. Just like CD and LS, like each one of those is a program. You can see that by, you can say which LS, and it tells you. Oh, well, that's a. I guess you can write them as programs, or they can be bash. We can see that GNZip is a program, and if you use file, it tells you it's a shell script, so we can actually read what GNZip does. Basically, it uncompresses files, and and it actually just calls the binary gzip. So it's not very interesting. Okay, so we unpack that. Oops, I forgot to check my checksum. Because these checksums are the checksums of the compressed files. Oh well, I won't bother with it for now, but you can if you want to. Then we have the FOSTA. Now we have the FOSTA, but we need to create basically pre-computed indices. So for whatever mapping program you want to use, you need to basically pre-process the genome. And so I like to make a directory for each different program we're going to use, in this case star. So like if you look at another example, like I have all these ribosomal sequences, you can see here that we have basically I think I forget which one I used. I think I truncated these. Oh yeah, into this file. And then here we have bowtie 2, BWA snap star, and another version of snap for different parameters. And so inside of each of these folders is like that program's pre-processed output from this FOSTA file. Okay, so looking back here, we go into star, and then at this point, just depending on the program, you need to follow the documentation. And so for star, then they have the downloads here and the manual. I'm sure I've already downloaded it, but I just download it again every time we need it. Then it says right here how to generate a genome. It's basically star, run mode genome generate. So I'm just going to actually just copy this. Oops. Oh, actually, so, okay, yeah, we'll need to do this as well. We need to get the annotation, and we can go without it for now, but we're going to need it eventually, and it helps the program since it's a gapped aligner. So, unfortunately, this is usually the hard part, is finding. Do y'all know, or do y'all have any idea of annotations for the maize genome?
I know we can search for chromosome numbers and these ID numbers, and they'll pop out predicted or sometimes confirmed.
So what we need is we need them all together in one file. The two most common formats are GFF and GTF.
I think, let's see, at least the iPlant thing has all the GFFs and GTFs.
Where's that?
It's that iPlant collaborative website I'm on with. Basically, it turns all the text work, the text-based stuff for bioinformatics stuff in GUI format, but it's down right now, it looks like.
Oh, it's the website's down?
Yeah, the website's down.
Oh,
It
okay.
has a whole database of like common plants and stuff actually. Wait a minute, I think I've done
So here it looks like, here's the genome assembly, and here's the gene annotation. And so here's the GFF3 form. So we want the full thing, which is here. Copy that. wget. And it has to match. Where do I want to put that? I guess I'll just put it here. Yeah, it has to match your genome version, which, because if it's saying there's a gene at this chromosome, this location, and the locations are skewed from one reference to another, then obviously it's important that you use the right annotation matching the genome reference you're using. Then unpack that. Okay. Yeah, 2 gigs, that's more like it. Okay, so let's go back to star, and then go back to the documentation, copy this thing here so we can modify it. Whoops. Oh, that's right. Yeah, it doesn't, the built-in Chrome PDF viewer doesn't let you copy, I don't think. Whoa. Rotate camera. It's star. Okay, yeah, I have it on the path. Minus run mode genome generate. Minus read files. Oh no, genome dir. And that's the. Oh, that's where I want it to spit out all the stuff. So that's right here. And the read files, no, the genome is 1UP ZMAs 21 pathogen. And so if you have multiple chromosomes, you can do it like this, or just all in one. Minus one thread, and we'll do 30. Yeah.
What does that last option mean?
So that tells the program how many cores it can use. And since with hyperthreading on this machine we have 32 cores, I give it 30 to give me a little bit of overhead still for moving around and stuff like that. And this, I think this usually only takes a few minutes.
I'm going to touch it.
And at this point, okay, so at this point we can make sure we have our original file. So I think, let me pull up that email from a long time ago. It's probably, no. Do either of y'all have the link of the
FASTQ?
What's it called? Yeah, the FASTQ files. I don't actually need it because I don't need to download them again, but I was just gonna kind of point out everything on there.
Here, I'll email it to you right now.
Oh, okay. Or you can just put it in the chat here.
I think it.
Okay, thanks. Yeah, so here, you'll probably, yeah, just stop me if y'all know all this stuff and you want me to just skip it, but, I mean, that's the FASTQ file. That's basically what they couldn't demultiplex. There's a little spreadsheet showing some numbers of reads and stuff that, you know, are in each of the groups that they consider. And then here are the FASTQC files generated for each undemultiplexed or demultiplexed group of samples. And then these are the these correspond to these here. And it's split across 2 lanes, which in this case, y'all didn't duplicate lanes, right? Yeah, so sometimes you'll spread data across multiple lanes and you need to combine it back together. But you don't need to do that here. Oh, they even give you the little wget command. So have y'all downloaded the data and started playing with it yet?
Yeah. So it's not just
Here, Danny, can you go and mute your mic if you're not talking, and just unmute it when you talk? Okay, cool, thanks. So have y'all started playing with the data at all?
I know we've gotten some information from it, but I personally haven't.
Okay. So yeah, I've already downloaded the data here in where was that? Yeah, Colomiets. Oops. Colomiets. Okay. And there's the 2 tar files, and then I unpacked both of them and it opened them up into this TLVR folder. Oops. And there's lots and lots of stuff in here. Most of it you won't use unless you really want to go like scrounge around for every last little bit of data. Then you usually won't use the pre-filtered or adapter found because those are, you know, first of all, there's not as much in there. For instance, like for this 7R5 sample, you know, there's 800 megs of regular FASTQ and only 44 megs of pre-filtered and 2.5 of adapter found. So, you know, even if you were to use both of those, it's only adding, you know, 5% or something like that. So I never really bother with it, except in rare cases. Okay, it's still sorting. And then I believe, let's see, I'm trying to think. Yeah, so these here are zipped rather than gzipped, which zipping is not as common in the Linux world as gzip and BZ2 are the 2 most common ones. But you can still just work with zips. It's a slightly different format, and so if you just go to the man page of zip and then read this, what you can do with it is, well, you know, when you're first starting off, you might want to just go and uncompress everything if you have the space. The problem is that'll use a lot of space, and especially on the cluster, I don't know how much quota Cronty gives you, and so you may run out of space before you are able to unpack everything. But that's okay because you don't need to unpack everything because you can actually just most of the time stream it into the program you're working with and then stream it out back into some compressed format so that you're never actually working with the uncompressed files, you know, all at once. And so to do that for zip, I'm trying to remember the command line. I think it's...
Is that cluster a free computer that we can log into, or?
Yes. Yeah, you just need to request a you need to request an account, Here, I can forward you the email that will let you do that. Threadgill. Here. Yeah, so the WSGI is an organization that, and they even, oops, I want to forward this to Eli and Danny. Okay. And so they have lots of resources, mainly for us, the computing cluster, but they also have, programs and databases and actual physical instruments you can use as well. So, and then Kranti, who runs the cluster, is also available to consulting and stuff like that through this WSGI Promise system. So that's good. And what was that zip? Let me just look. So, as part of our lab, we actually have a set of scripts that we use to do this sequencing pipeline, basically. And I'm not covering them here because I guess we haven't we haven't actually released them openly yet. But also, I think it's best to just start off your first times doing it by hand. Oh yeah, zip. Unzip minus P. Okay. So if we unzip minus P. Yeah. So minus P extracts files not onto the hard drive, but to standard out, which basically means kind of like printing it out to screen. But if we do it a certain way, then it doesn't print out to screen. It actually can get captured by a program, and the program in such a way that the program thinks it's a file. And so that's trying to think what I should show you next. First of all, is all this making sense so far?
Well, I'm trying to follow along.
Oh, okay. Well, stop me, or here's a perfect time. Start asking questions.
Okay, how?
Or even clarifications. Yeah.
So this black box that's the terminal can be accessed from that email linkage after requesting an account.
So right here, I'm on my laptop, but I'm remotely logged into a machine that is owned by my PI or my advisor, which is very powerful. Lots of cores, memory, space. Although it doesn't actually have to be that powerful. Like most modern desktops, if you have enough free space on your hard drive, well, RAM is sometimes a problem, but so it depends on the program. But you can sometimes get by with 4 to 8 gigs, but with Star specifically, you need something more like 64 gigs or something like that. And actually, I'm not sure if this is even going to work in Star because Star has some. Sorry, I was getting into details. Yeah, so then to answer your question, by that email I sent you, you can request an account on another server, basically, that will allow you to log in. So this black box is called the terminal. And so if you launch a terminal or you download a terminal program in Windows, are you in Windows or Mac or?
Yes, Windows.
Okay, so yeah, you'll want to download something called PuTTY. It's basically an SSH client. They have a horrible website, but yeah, you basically just download this, and then it'll allow you to just type in the server name and your username and password, and then click connect, and it'll open up basically a terminal like this logged into that system. And then once you do that, then you'll be in a Linux environment, and you can start, basically it's like it's a little window into that system, and you can download stuff to that machine and run things on that machine. So none of this is running on my computer, for instance. Like I have a little dinky laptop that can barely handle this Hangout, and so it would not be able to do this alignment at all. And so that's how I'm doing it, is because I'm remotely logged in. Does that make sense?
Yes, yes.
Okay. What other questions?
And
Or here, I'll ask you questions. Did you understand, what the reference genome is and what the annotations are?
so reference genome is like your sequences that are arranged by chromosome, and the annotations would be the labels that would tell the program where are the genes starting and stopping.
Yeah, exactly. So here, let's look at it. So less is another Linux program that just lets you look at files, text files. Because I could open it up in a text editor, but that would be, overkill. And so less just lets me, quickly look at these huge files. Because this is a 2 GB text file, and if you try to open that up in, Notepad or something, it'll blow up. But less just lets you kind of look at little pieces at a time and scroll up and down like I'm doing here. And so yeah, this is just a FOSTA file. It's basically saying, here I've got this a scaffold here, and it's only a few letters long, or a few base pairs. So I think they have all these scaffolds at the front. Huh. I'm surprised they have this many. Wonder if we search for chromosome. Okay, so here's, I think, chromosome 1, basically. And you can see it's just, really long. And then if we search for 2, it's a scaffold. Oh, there's chromosome 10, I think. And these ends are regions where it's unable to find the DNA. Or it doesn't know what the letter is, but it knows there's something there. Then this here is the annotation. Okay, still. And basically this is a weird. And so to look up what these columns are, you just say GFF or whatever, file format, and then click on like the first thing, and then it'll tell you what all the fields are. So the first one is the name of the chromosome or scaffold. Then it's the source, where this the name of the program or the institution. Then the feature type, whether it's a gene or a variation, or they can have lots of different things. Coding sequence, start codon, stuff like that. Start position of the feature, end position, a score, a strand, a frame, a tribute. But the most important are the first ones. So these are repeat regions. Here's a coding sequence, 3' untranslated unit, transcript. I'm definitely confused at the chromosome numbers, because normally this says right here, this first one is like chromosome 1, or chromosome 2. And the fact that there's no chromosome numbers is strange. But maybe that's Ensemble's convention, maybe that's a Corn convention. I honestly don't know. Oh, wait. Okay, it's still going. Do you understand what we're doing right now here to the reference genome?
No, Your
We have annotations to the correct spot.
Wait, say that again?
Are we, are we, right now, are we trying to map the annotations to the correct spot in the genome?
Close. And actually, that's a good point. I forgot to give it the annotation here. See, I just told it, uh, you know, run in generate mode, put the output right here, and then here's the FASTA file, and use 30 cores. But if you look in the documentation over here, it says, yeah, if you have a splice junctions database, you can have more accurate mappings, but you need to basically give it. Wait, yeah. When you're generating the genome, you need to give it this extra flag here saying this is the file name with the annotations in it. And it can either be in GTF or GFF3 format, which, as you can see, this is in GFF3 format. So I should have done that, but I'm not going to stop it and rerun it since this is just a demo. Man, has it already been 48 minutes? So do y'all need to leave at a certain time? I'm also busy, so I don't want this to run too long, but I don't want to overwhelm you or not get you anywhere to get started either.
So some good homework would be to make an account, do the putty, learn how to use putty,
right.
learn how to use star.
Yeah, that's so that's one thing is star is just one of the alignment programs. Really, the granddaddy of all of them, or not the granddaddy, but I don't want to call it the gold standard either. Whoops, that's not the right one. There it is. Is Top Hat. Top Hat is basically what everyone uses, and it doesn't need as much RAM, but it's also much slower than Star, which is why we've been using Star. But so one thing you might want to do is go to something like BioStars, or even just Google, and say something like where's search here? Live search. Zia Maze. Mapping or something. No results. Corn mapping. Surprised there's not. Wait, is it?
No E.
Well, oh, yeah, it is that maze. Okay, good. Mapping. Oops. I guess I didn't want to. Let's just try Google. Maze mapping genome. Or I guess RNA-Seq could be. I'm just wondering if there's in the corn community, is there, everyone's centered around one mapper or something like that. It doesn't look like it. Although, I've only done 2 searches, so who knows. And then this is another, really popular one, as you probably saw in my YouTube video. And so this process, or this tool here, STAR, some people call it alignment because you've taken these reads and you're aligning them along the gene. Some people call it mapping, so you're mapping the read onto the genome. And it's, I don't think there's really any difference either way. I mean, they're just 2 different terms for the same thing. And you can also do de novo alignment, which means you have all these small reads, but you don't have a reference genome. And that's where you would actually be creating the reference from your pieces, which that's a very different and much harder problem. So it's good we don't have to worry about that here.
Just wondering if it's worth it for us to try to convince our boss to convince the department head to buy a computer for this, or is it just better to log into that website?
I would honestly, I would honestly just use WSGI because it's there, it's free, and before you really know what you need, then you can easily waste money in one direction or another. And this is a free resource that's available, and lots of people use it for these exact purposes. So, we have our own. Because we do so much of it, and we're actually part of a center now, so we actually do other people's data as well. So it makes sense for us, but in general, you want to leverage as much shared resources as you can until you feel the pain of not having your own. If you constantly are having to move data back and forth because for whatever reason, or so not yet, I would say. But I mean, if he's got money sitting around and y'all are going to be doing a lot of this, then it might make sense to invest in that. But definitely start off on just the WSGI, and if you are running into the barriers, then at that point, then you can reevaluate. Oh, let me just show you here how to use Top Hat too. So I'm in this folder, like I showed you, and we'll make a Top Hat folder. Or actually, wait, how did I do this? So let me. So Top Hat is actually kind of a wrapper around another tool called Bowtie. Where was it? There it is. Okay, Top Hat is a read mapper for RNA-Seq, as you can see right here. But Bowtie was written earlier than Top Hat. It was like the first program they wrote, and it's just a read aligner, and so it doesn't handle gapped reads. And so basically, they then wrote a little wrapper around Bowtie that handles the gapped reads, and then they called that Top Hat. So we're actually, even though we're going to use Top Hat on this because it's RNA-Seq, we actually need to make a Bowtie 2 alignment. So we'll say make dir Bowtie 2. Go in that folder, and then now we check their documentation for Bowtie. Actually, Bowtie 1 and Bowtie 2 have different websites. So this is Bowtie 1 website, and you can see down here Bowtie 2's website. So that's something that's kind of confusing. And then the manuals right here. Then they have a really nice manual. I'd recommend, just especially for just starting out, just read the whole manual, very first. Even though you might not understand what it's talking about, some of the parts, then it'll jog your memory later on, and you'll need to read it once or twice more once you understand more stuff. But so here they say how to obtain Bowtie 2. It'll already be installed, probably on WSGI. Oh, that's another thing about Star is Star may not be installed on WSGI, so you might have to install it yourself, which actually you would just download it, I think. Does it have binaries? So one thing about software installation in Linux, this is an important point, is if you install Linux on your laptop or you have a workstation or something like that, then let me show you here. So here, this is on my personal laptop right here. And so if I wanted to install some software, then usually I would use a package manager like Yum. Say Yum search, a program like Top Hat or something. And since there's so much open source software, they've put it all into these online databases. Actually, I don't know if they have Top Hat. They might have Bowtie. And basically, let me see how they have Bowtie. Oh, I know they have SAM tools. That's another program you'll end up using. And so then, to install it on my machine, I can just say sudo yum install SAM tools. And then it'll go download the software and then unpack it on the system and then make it so I can run it. So it's now SAM tools, it runs. But if you noticed, I had to use this sudo command, which I don't know if you saw on linuxcommandline.org or commandline.org, if they have it, that sudo means super user do. So do something that can or will affect the whole system. And so since it's my personal machine, I have that power. But on the WSGI cluster, you won't have that power because you're going to just be one user among many, and only Cronty has the sudo powers, basically. And so you, as just a user, you live in your home directory, home, and then your username. And everything in that directory, you can do, like stuff to write, read, delete, make new directories, whatever. But anything outside of that directory, you can't. So you can install star inside of your home directory, but you can't install it system-wide. So you won't be able to do anything like yum install or apt-get install or any of those package managers. Does that make sense?
Yes, yes. And when you go to use a manual, that's what they'll recommend how to install it through.
Possibly. Some might say that. It depends on the tool. So, like star is a new enough tool and not popular enough that they know that it won't be in the repositories, and so they'll they won't even use the package manager because they know they're not in any of the repositories. So yeah, if you look in the manual for star, it'll say basically grab the source code. This is a tar GZ. So this is a tar is just a tar is a way to turn a directory into a file. And then once it's a file, you can then GZ it. So it's zip, you can either zip a single file or a folder, and you end up with a zip file. But here, gzip only works on files, and so to do a directory, you have to use tar. Does that make sense?
Yeah.
Okay. Sorry, I'm throwing so much at you, but I figure throw a lot at you, and then some might not stick, but maybe, and you can watch this video again later too once you are more on your feet. Yeah, Cronty's upgrading the WSGI cluster. Actually, he's starting that today, and so you won't be able to log into the cluster this weekend, I think. And he might not even make you an account this weekend, so since he's busy with the upgrade, so that's something to keep in mind. Yeah, so he says here, download the source from his website, and then go into the folder or unpack it. Go into the folder and then run make. And what make will do is it'll actually compile the source code into the final program, the binary. But you might not have to do that because if you download a pre-compile executable, then he's already done that compilation for you. And that should work on most Linux machines, so you won't have to do that. And here he actually gives instructions for installing the prerequisite programs if you are going to try to compile the software yourself. Sorry I'm throwing so much at you. I can't imagine how overwhelming this must be, because I've spent years, figuring all this stuff out on my own. And oh, okay, here we are. Oh, okay. So now let's use bow tie real quick and make another reference. So this is the documentation here for bow tie 2. And it talks about how it aligns, which this is very important for you to understand, how it's actually going to do the alignment process. Because then it'll make sense, when it talks about the error rates and, how many mismatches and gaps and stuff like that. I just don't necessarily read all of it. I don't know if I've ever read all of it. It's pretty extensive. So where am I trying to go here? I wanted to do, oddly enough, it's kind of at the end where what we want. We went to the bowtie 2 build indexer. So this builds a bowtie index from a FASTA file, which is what we want. And here is what we were looking for. This is how to run the bowtie 2 build program. So it's basically just bowtie 2, then your FASTA sequence, and then what is that? Oh, the base name. So you can just make up a name here. I think I call mine genome. What do I call mine? IMO bowtie 2. Yeah, genome. So here we can say bowtie 2 build, then the ZMAs. Oops. Oh, and let's look and see if it takes an annotation. I don't think so. Oh no, because it's bowtie. Yeah. And remember, bowtie doesn't do RNA-Seq, right? This is just kind of setting up the bowtie program on an arbitrary FASTA file. Genome. And you can see here our star index completed successfully. And so if we look here, it basically produces all this stuff that we don't care about what it is. And some of it's large, 2 gigs, 1.5 gigs. And it does this to make the problem feasible, right? Because if you took each of your reads and you scanned it across the entire genome looking for an alignment match, that would take forever. And so these programs are highly efficient, using very exotic kind of algorithms, data structures, in order to make this process possible. But in order to do this, it has to do with all this pre-processing only once, right? So once you do this, then you're done. You don't have to do it anymore. Oh, and another thing, let me go ahead and show you. Let me log into WSGI. Oh, that's right. It's not going to work because he's upgrading it. Well, that was a good idea, but it's not going to work. Yeah, what was his email? I think he just said he's starting. Oh, no, he says it's done. Okay, that's good. So maybe you can get one. So why can't I log in? Let me try doing it from here. Oh, okay, there we go. Yeah, so on here, he has some, since everybody's doing genomics on this cluster, he actually has some shared resources like reference genome indexes. So you may not even have to build some of these. Like he may have a maze one. Maybe not for maze because it's not as... where would those be? Well, that's something you can look at once you... oops. No. Dang.
I think it's in that rough underscore FASTA stuff.
Oh, yeah. Okay. Yeah, good point. Ref indexes bow tie 2. Yeah, I don't see maze. I think it's bull. You'll probably know more of these. Horse, maybe. Human, mouse, rat. Okay. But you could probably ask him, and I bet he would be happy to make a maze one for you. And yeah, so in here. Oh, okay, that's still working.
What would that be? That would be a maze genome upload?
Right. So here he has, since it's a shared resource, and it would be silly for everyone to have a copy of their organism's reference in their home directory, right? Because that'd waste a lot of space, because lots of people would have home or human references, for instance, and the indexes. Then he goes ahead and makes those publicly available to everyone. And so here are the annotations. And so, if we look at human, then you can see the GTF reference annotation here. And he also has the FASTA for the reference for human HG. There's the FASTA file. And then indexes for bow tie 2. Yeah, and so this is what we're going to see bow tie 2 generate for us, except for maze rather than for human.
I have a question about
the
WSGI itself. I read the wiki and it says for intensive jobs they ask you to use the UGE stuff like keylogin. Is that, I'm not really sure how that works. Is that like you keylogin, like request the node, and then you submit a job by using a bash script?
Right.
Is that okay?
So keylogin is the easiest. That's basically saying, or first of all, Eli, since you don't know anything about WSGI cluster, I'm assuming, basically the cluster is designed like a star, in that when you log into it, like I am here, you log into what's called the head node. And the head node is the entry point, and then you're not supposed to do anything computationally intense on the head node, because if you did, it would slow everybody down, just getting to their data or whatever. And so to do anything intense, like moving data or computing, you're supposed to actually move to one of the compute nodes. But you aren't supposed to access the nodes directly, or first of all, you can't putty in directly from your computer, because it's connected like a star, so you have to go through the head node. And you don't SSH into the compute nodes from the head node. You use a management system called the grid engine, basically is what it's called. And so you ask it, I want to log into a compute node. And so you say qlogin. That gives you basically a session on one of the compute nodes that isn't busy. And the reason they do this is for balancing loads across computers evenly and stuff like that. And so now I'm on a compute node, as you can see right here. And so now I can move my files around and do computations and stuff like that. And as you'll see here, my home directory, which is where I am, home binary bana, here on the compute node, do I need to get that?
It got it.
Okay. My home directory on my compute node is the same as on the head node, and it does that because it's a network file system. So your home directory, the files and stuff, is not actually on either the head node or the compute node. It's actually a network file system, so it looks the same each place. So here, if I touch a file saying, hey, now there's a file named hey on the head node, and if I go into a compute node and I look, then there's also a file named hey, because they're both looking at the same system or file system, basically. Does that make sense?
Yes, yes.
Okay, so that's one confusion. The other confusion is Cronty's very particular, and there's lots of quotas about where's the promise system? Oh, it's promise. Let me just say HG. There it is. Okay, once you get an account, you'll have access to this, and you can. Oh, I gotta log in. Yeah. And then you can go to the compute cluster project, and then the wiki, and then using the compute cluster and guidelines. Oh, and he also has a Galaxy one. And I'd recommend reading all of this. It's kind of hard to read, but basically you're supposed to put your big files in a special directory, not in your home directory necessarily. He has blast databases. Yeah, this isn't the easiest thing to read, but I'd recommend trying to get through it as much as you can. Okay, so that's Q login. Does that make sense? Or how much questions?
No, I think I understand.
Okay. Okay. Here's an example of some quotas I have. So on this file system, which I think is my home directory, I'm only allowed 8 gigabytes, for instance, which isn't very much. That's a couple of files for genome applications. But as you'll read here, the quota on the storage. Yeah, 2 terabytes per user in the data directory. So here, if I look in data binary BANA, then I only have a test file. And I think if I look at my quota there, how do I? No, that's not what I want. But anyway, there's some way to figure out what your quota is on that other one as well. Yeah, so you have a 2 terabyte quota there. So that's where you put your files. Oh, and the way you move files back and forth is probably using something, what's it called? FileZilla.
FileZilla.
Yeah, okay, you've heard of that? Okay, so by default, he doesn't have FTP set up, I don't think, but FileZilla can use something called SFTP. SFTP, which is basically file transfer over SSH, and I think that works. For example, here if I'm on my laptop and I want to push this little text file over, I can say secure copy that file to WSGI and then my home directory, and then it would work if I could access it from here right now, and for some reason it's broken. But I could do it from this computer probably. But anyway, it's not too important. Yeah, so FileZilla will just give you a nice little graphical program where you can copy those things over. Although actually, what you want to do, since y'all have the data already right on the Charlie Johnson server, you just would you'd go onto WSGI into your data folder. Oops. And then you'd say wget huge file, basically, and then it would put it straight into your, 2 terabyte quota directory. Does that make sense?
Yes. Yes.
Okay. Okay. So what I was going to show you. Oops. Because you, Danny, asked about qsub. So okay, here. Let me tell you the bad parts here. Let's say I say, something like run really long job here, and it's sitting there running and running, and then I close my window, or maybe I get disconnected from the internet or something like that. Just based on what I've done here, that will actually kill the job, I believe. Unless QLogin does some fancy stuff. Generally, when your SSH connection dies, then your job will die as well. Obviously, that's not a good thing, right? The other bad thing is that, let's say the cluster is full, right? And then you say QLogin, and you get put in a waiting queue, and it doesn't run until next Thursday or something like that. That generally doesn't happen because there's not enough people using WSGI yet. But basically, that's why there's this job submission system. So qsub is a job submission system which says, okay, I'm going to write up my job in a script or something like that. And then once a computer or a group of computers becomes available, then run my job and then tell me when it's done or something like that. And so you can read all about qsub and how to run it. And actually, Cronty has a pretty good write-up here on how to use qsub. And don't worry about all these things. There's lots of things you can also do, like deleting your jobs and checking on your jobs and looking at your previous jobs and stuff like that. But mainly, you just want to use qsub. And here's a little example here too. So basically, if you write a little script saying, process my data, then you just say qsub and then the name of your script, which all a script is, Eli, if you don't, or I guess you were going through Linux command line, so you might know. Well, both of you, if you don't know, a script is basically just a list of commands, one by one. And you can even do little for loops too, saying, for each thing for each file in my directory, then do the alignment. So actually, I should show you that. Yeah, let's do a little test. Okay, and this will be the last bit, and then y'all can go do homework, and we'll reconvene, whenever. So let's make a bunch of phony data in my data folder here. I'll delete test. Okay, so I'm just gonna say for INC 10, there, we'll say 30. Do touch data I done. Okay, so now I have 30 fake data files here. Imagine these are FASTQ files. Actually, let's do that. Okay, for INC touch.fastq. Okay, now we have 30 FASTQ files. They're completely empty.
What's the little O that you?
Oh yeah. So I've aliased O and O to be just ls and ls minus l, because you use those commands so much that in the keyboard I use both the L and the S and the enter are all on my pinky, and so ls enter is really hard, but O enter is really fast, and so I just aliased it, which alias just basically means shortcut.
Gotcha.
So whenever I type O, it just does the same exact thing as ls, and OO is long list, so I can see the sizes here. Actually, and just one little Oh, I don't have it here. Yeah. Anyway, so that's our data. We'll go back to our home directory. This little tilde means home directory. And we'll make a little script, say process my seq data.sh. And here, if you go through the commandline.org guide, it'll talk about writing scripts. This is basically just a little thing telling the Linux command that this is a shell script. And then I can do lots of stuff here. I can say echo hello. I can, copy a file from hey to hey2. I can also go into my data folder, and then now for each file in this directory, I want to actually, I forget how to write it. I think this is how. And then I want to process this file. And so I'm actually let's make a directory in my home directory called results here. Then so for every file that I'm going to process the file, which since they're all empty files at this point, then I'm just going to say echo processed file F, and then I'd like to be able to see the results from that, or something like that. So I'm going to output the results into my home directory, results, then f.results. And then I think I say done. Do I need a semicolon? I forget how to write for loops on multiple lines in Bash. Oh, I need a do. That's what I need. Do. Okay. I don't think I need that one. Or that one. Okay, so let's try that. So then I qsub this proc seq data, and then it says it's submitted, and then you can see that it's running right here with qstatus. So I think I forget how to tell if it's running or not. You can find that information in here. He describes that more. And there's tons of options you can put to qsub, like here's my email address, email me when I'm done, save all the error output here and the regular output here, stuff like that. Then we can see, we can actually see it's already done stuff. It copied hey to hey2. It made this results folder, or did it make a results folder? Oh, it put the results folder in there. So if we look in the results folder, it's empty. So I must have messed it up. And so that's actually one of the bad parts is, it's like, how do I figure out what went wrong? And right here you can see that because you can't see the output, do you need to answer the phone?
No, we're good.
Okay. So when you qsub something, you can't see what happened, right? Because it went and ran on some other computer. Heck, it might have run. It might not run until tomorrow because the system might be bogged down, and so we're not scheduled to run until tomorrow. And so once it finally does run, it saves the error output and the regular output, standard out and standard error, if you learn about those terms, into here. And if we look at those, proc seq data she, then we can see that it had an error, it looks like for each one of our things. And it said no such file existed at home binary banner results. Oh, okay. So there was if we go back to our script here, we can see that here we first changed directory, then we made a directory, or then we made the results directory, and then we tried to save it to home results. So that was the problem. This should have been above here. So if we Save that. Get rid of Hey2 and get rid of data binary banana results. But yeah, I have to do my center. Okay. Then now we can Q-sub it again. Where am I? Product data. And then once it's done, we can just watch it here. You can actually say watch QStat, and it'll run it every 2 seconds. And then whenever it disappears, that means it's done running. I believe is what that means. Yeah, okay, so now it's gone. So now we can see our results folder, and it's got all these results in it, and we can just look inside of one of them. Data1.fastq.results. And you can see we processed file data1. Does that make sense?
Yeah, there you can use it. No problem.
Hello?
Yes.
Okay. And this still isn't done, but and so this is a way that, you know, even if your connection failed, you know, your job would keep going because it's handled by this submission system, basically. But this is, you know, like I said earlier, this is the kind of underbelly of this processing, and you may want to first start out on the Galaxy, you know, because here he describes using FileZilla, so definitely check this out. Here's how to use FileZilla with the WSGI and your username. Then you copy your data. Oh, well, it doesn't actually describe how to use Galaxy, but there's actually quite a bit of good documentation on the Galaxy page itself. Oh, and you won't be able to access this. You have to access WSGI from on campus or through the VPN. Okay, well, I think that's probably enough to overwhelm you. So let's switch to homework at this point. Or do you have any final questions?
No, I think that's a very good foundation.
Okay, great. And I'll try to send you the link to this recorded video so you can watch it again if you want, or look up something. And then actually from this point, you know, where we left off here, I basically just built the index. Then you can follow my RNA-Seq tutorial to do the next step, which is actually the mapping. And you know, if you want to get that far, or you know, see how far you can get, and then maybe we're gonna have one more session about, you know, once you've kind of come to grips. But I am leaving for vacation right after finals, so I won't be back until May 15th, I think. Yeah. So mid-May. And so yeah, I guess we'll figure out a time or something if y'all need more help or whatever. Okay, does that sound good?
Sounds great.
And
Yes,
feel free
thank
to
you.
email me questions or anything like that too.
Definitely. We'll do.
Okay. Well, y'all take care, guys. Good luck on finals if you have any.
Yeah, thank you.
Thank you.
Okay, bye.